About 25% of the TJ River, such as it is in San Diego, and sewage is regularly entering the watershed. It moves downstream, causes flooding damage, and it's a huge health hazard. And we have terribly different, totally different regulation on both sides of the border and different issues. It's a problem. About 2.8 million people live alongside the river, including 83,000 on the US side. So that is a massive amount of people who have to deal with the pollution that's happening there. Runoff is contaminating our beaches. Imperial Beach is closed like 200 days of the year, and now it's starting to hit the news, right? So now we have Padilla, Schiff, Booker, Vargas, Peters. They're introducing a Water Quality Act. I don't know if it'll go anywhere with the current administration. Ontario Lost and Reamer is really putting time into this, and we're getting to 1,000 days. I don't know what the time frame is. 1,000 days of beach closure. Now that rich people are starting to be affected, maybe some action will happen, right? Okay, so what is the TJ River and how dangerous is this? This is supposed to be animated, so I don't know if my animation is working. So what we need, at least this is what I'm focusing on, is a method that can simultaneously identify, to try to figure out how dangerous it is, pathogens, antibiotic resistance, and the sources of contamination, right? So hopefully, does none of the animation work? Okay, it's working again. All right, so here is an example, electron micrograph, was it SEM? No, TM, anyway. A bunch of bugs, right? And they colored it to make it look, but they kind of look the same. So we're going to use a method where we can study them all at the same time, and I'm going to call it Mr. Metagenomics to the rescue. Metagenomics is basically a way to look at all the DNA of all the organisms exactly at the same time, right? It's a lot of data. All right, so why do we need a method like this? Well, microbes are small. This is a tip of a pin, you know, angels dancing on a pin. Well, this is bacillus on a pin. They are extremely tiny. This is a shower curtain biofilm. I don't know about you. All of these dots are bacteria, but they all kind of look the same to me. They're really hard to tell apart. They don't have feathers and wings and claws and all this kind of stuff. They have a couple of shapes. And another problem is what we knew for the first 100 years of microbiology is what we could grow in a lab, right? So you have labs. You've probably seen the Petri plates. Excellent. Except you can't grow most of what's out there. And we learned this relatively recently, and it makes sense, right? So maybe the stuff needs sulfur. You don't have sulfur. Maybe it grows at 90 degrees Celsius. Maybe it needs another organism to live, right? So that's the problem. And how are we going to solve it? All right, so to solve it, I'm going to give, so what do we do? So I'm going to give my giant, this is what we're going to do. I'm getting my analogy. And my analogy is to think like a giant alien, right? Resident alien. So this is kind of like, if you're a giant alien, this is what the Earth looks like to you. This is kind of like our microbial world, right? So you're so big, you just see a lot of blobs. Oh, there's some green stuff here and some brown stuff there and some wet stuff there. And it's like, it's all so small, how are you going to study it? Well, let's think like a giant alien. So the alien goes in, this like says, hey, this part in the Yucatan looks kind of interesting, but I can't see it. I don't know what it is. So what am I going to do? So you take it, so let's do old school microbiology. By the way, old school microbiology is great. I love old school microbiology, very important, but it's not going to get everything. So in old school microbiology, you're going to go look at that spot, you're a giant alien, you're going to take a swab, and you're going to go down to that spot, and you're going to scoop up a bunch of rainforest, right? So you scoop up a bunch of rainforest, right? And then you want to say, well, how can I grow it? So let's give it a growth media. So growth media for the giant alien is to put it in a cage and feed it a bunch of papayas, okay? I mean, it's silly. It looks silly, but it's kind of silly to give it seaweed. And that's what we do on a plate. So we give it that. What is it going to grow? What do you think? Fruit flies? Yeah? Fungi? Right? I don't know, some beetles maybe. But it's not going to grow jaguars. It's not going to grow figs. It's not going to grow any of those stuff. All right? So you can get some stuff, and it does come from the jungle, and it does eat papayas. But what about the rest of it? Well, let's try another approach, where instead of trying to grow it, we put it in a blender. All right? We put it in a blender. We grind all the organisms up, and we extract out its DNA. So here's the DNA in a little tube. And you can say the great thing about DNA is we know how it behaves chemically. It's a long, negatively charged molecule. It's easy to purify. It all kind of acts the same way chemically. But I can tell the DNA from a jaguar and a fig tree totally apart. Super easy, right? OK, so that's my analogy. And this is kind of how it works. We can get all the DNA from all the organisms all at once. So here's my microbial community. Inside all the cells is a circular thing of DNA. And I'm going to bust all the cells open, chemically or physically by shaking them. I'm going to extract all the DNA out. This is my DNA molecules, which readily turns into letters in a computer, right? So there's sequences is very easy. There's nucleotide bases, and there's only 4 different kinds of letters. It's the most boring paragraph you will ever read. But you get more and more and more of that data. So get all the chemicals. Then the data comes out looking something like this, and you can just eyeball it and see that pattern is different from that pattern, like different sentences in a book. So each one of them is a different organism. Use the patterns to determine who's there and maybe even what they're doing. And all of those are all the cells, all the different kinds of cells. And you can even figure out relatively how many there are, right? So it's really useful. OK, so let's go to the sample collection. So this is where we worked on it. The sample was, we started at the border. This is just one of the studies. Took 6 samples here over a bunch of different days, and also down at the river about the same time. And these are the dates. We extracted the DNA, purified it. Just a little context. A bunch of these days there was rain, so we can get some flow going. And 2 of the days, we know that there were actually big sewage leaks. That'll come back later, all right? So we knew there were overflows. All right, so what do we find when we look at this stuff? So I can't show you all the data. It's a massive amount of data. But here's a list of the top 10 microbes. And they don't mean anything to you, but I'll tell you what they do. These are from Radio Club, which is at the border. And these are from Boca Rio down at the ocean. So the stuff at the border is coming down to the ocean. Obviously, it's smaller numbers. It's becoming more dilute, but it is there. So what do we find? Well, great. We find Comomonas, common and activated sludge. Aliarchobacteria, that's a fecal pathogen. It's a growing problem. Antibiotic resistance and Acinetobacter johnsonii, which is a known problem in hospitals. It's antibiotic resistance, activated sludge, a urine pathogen. We even find these viruses that we know are in fecal matter. And every time we do it, there seems to be new kinds of organisms. We can also look at fecal markers. We can give them directly from the sequence data. So we can use things like E. coli to measure it. Usually you have to plate it, but we see them directly in the DNA sequences. So in Boca Rio, in Radio Club near the border, we got a lot more than down here. And then we used the crassphage virus, which was discovered here. The genome was put here as a marker of fecal material from a guy named Rob Edwards. And you can see that it's higher there and lower in the ocean. And then we have antibiotic resistance markers. Again, much higher on the border, much lower at the ocean, but they're there. They're flowing downstream. In fact, we have almost a linear line as you get farther and farther away from the border. This is near the border, a little farther away. This is another study, all the way down to the ocean. So all these antibiotic resistant markers are getting down there. OK, last thing is my source tracker results. So together with some other people, I invented this program software called Source Tracker. We could tell you the origin, environmental origins of microbes from a given sample. So these are all the samples from different days. Radio Club from the border, and Boca Rio from the ocean. And here's the places that we predict each of the environments to come from. So what proportion comes from wastewater, which is like sludge? What proportions come from soil? We had 2 different ocean types, deep water and near ocean, gut, and freshwater. Gut would be like sewage, right? So what do we see? Well, it's pretty clear to the naked eye that gut is near the border, right? It's dominated by wastewater sludge and from sewage, right? As we go to the ocean, obviously we get lots more ocean bacteria, except for 2 days. Which days were those? That fit exactly what we're saying. So we're linking the overflow and bad days to major pollution events, and we can also say what organisms we have. And I will do one more thing that came out of this. It's really fun. It's not fun actually it's not fun. None of this is fun. I want this to go away. I don't want this ever to happen again. We can talk about solutions. But we were contacted by some wildlife biologists, and there were some dolphins that were having this terrible eye disease. I don't know if they died or what. And they said, hey, do you have this organism in your data set? And we did. So they had some disease that was affecting the dolphins. It was in our data. We can't effectively prove that, but where else would it come from? It was like a pathogen from sewage. So we know that they're infecting. And now we're working with all sorts of other wonderful people in the Health Link. These are my colleagues, NIH. But in the Health Link, we're also looking at, I don't know, the slide didn't work so well. we're continuing this study to look at how it's directly affecting the community. So it's one thing to be in the water. It's another thing, does it travel distances? Are people getting antibiotic markers? Are they getting the disease? And that's our next step. OK, thank you. Oh, sure. Well, you have to have sewage treatment, right? And they're just, you have to have a cross-border, and you have to massively scale up the sewage treatment plant. That would be #1 thing to do it. But that only solves it on our side of the border. There's 2.8 million people on the other side of the border who are living right next to it and are affected by it. So there's really a lot of politics going on here, of course. But Tijuana grew so fast and so rapidly from, we dumped corn on, you guys know this, in NAFTA, like we dumped corn on Mexico. That caused 1 million farmers to lose their lives. Where are they going to go? They went up to the border. Tijuana grew massively. There wasn't any infrastructure, right? And now we have an asylum problem, partly of our making. We have a drug problem, partly of our making. All these people coming onto the border, they have nowhere to go to the bathroom, right? That's a huge problem. The other problem is the rich people. It's not their rich people are a problem. It's just that the houses where they're, one of the big problems, they blame the people who don't have a place to go to the bathroom, but they dump their sewage right into the river, right? So you could work on that problem, try to get them good sewage treatment in the rich areas. In the other areas, we could find a way for people to actually have somewhere to go to the bathroom. I know people who are working on like really cheap, portable, compostable bathrooms. I mean, if we really wanted to, we could really take a stab at this. But it's going to take politics, it's going to take caring, and it's going to take binational working. And right now, we have an administration that doesn't seem to want to do that. So, but yeah, a bigger sewage treatment is the #1.
Looked like you started back in 2020. Were you also looking for coronavirus?
Oh, not in this study. That's an RNA virus. Yeah, people were looking for it, like in the air and the water, but you have to use a different method. So this is DNA only. We could, these are RNA viruses. You can convert RNA to DNA and then sequence the crap out of it, as we like to say. And that could happen. We definitely have some people in the health link are looking for, what is it, HPV? What's our collaborator? Oh, maybe you guys don't know. There's other viruses that they're targeting right now. Some of them are DNA, though. But yeah, there's a lot of other viruses. It's a little trickier. But yes, it's an absolutely extremely important thing to look at. Yes?
So when I heard you talk about some of the history that underlies people moving from one place to another and their population, lack of infrastructure, I almost hear a bit of anthropology in your work. And I'm wondering if that's something you have.
You mean in my opinion?
But for example, like, I'm thinking about collaboration.
Sure.
Right?
Yeah, yeah.
So when I was listening to you speak, especially looking at like the 2 like rainy days, talking about Coronado and movement, I was thinking of Jason P. DeLeon's work at UCLA. He runs something, a large-scale project to look at how people move from one place to another, almost using archaeological type methods to document what's happening. And I don't know that you would be able to apply this to the types of research that you're doing, but just a comment slash question in terms of thinking about different types of interdisciplinary implications.
Oh, I love it. I would say that the 2 areas that, so temporarily is the problem. Like we're just collecting it now. We don't know what happened in the past. And it's good to match those things up. But the 2 things we know are happening is massive migration patterns, people getting stuck at the border. That's happening very recently. I notice that each time I do a study, the microbes kind of change. Right? It's not totally static. I see the same ones, but other times I found just lots of E. coli. So, what's up with that? Why is it changing so fast? The other thing that's happening a lot was we do chemistry analysis. This isn't my work. This is Unha Ho and some other people, and they're finding loads of meth, right? So we're starting to see the drug war turn back on itself, and they're selling to local. I mean, it's just it's a disaster, right? It's a disaster that we're just ignoring and saying, oh, it's their problem. But I agree with you completely. If we knew those type of things, if we also knew more about the hospitals, I don't know, and farming and industry, like what is going on there, those are things that we really are just blind to, But it's a real shame, though. You imagine what that river must have been like, right? I mean, it's a sewage pit right now, but it must have been the most beautiful river flowing up through the border, trees and plants, and it's just raw sewage sometimes. It's hard to think about, you know. Other questions? It is on a depressing note, yeah. Okay. All right, I brought the house down. Okay, I did my job, but yeah, serious stuff. Thank you.